Today we discuss about the bacterial genetics that comes under microbial physiology metabolism, that is BVMLT 303. This topic come under unit 3. So in bacterial genetics, what's the main topic? That is conjugation, transformation and transduction. In this topic we see that how the bacterial genetics will transfer from one cell to another cell. So first of all we discuss about what is the bacterial genetics. You already know what is the genetics. Genetics is the study of heredity and variation. Actually it is the main study of the gene. Genetics is the main study of the gene. In genetics we study only about the gene, how they look, how they transfer from one cell to another cells, how they act as a heredity material, how the variation comes under the gene and they cause the mutation. This all properties are studied under the genetics. So the unit of heredity is the gene. What is gene? Actually gene is defined by Mendel in Mendelian genetics, but they not called them gene. Mendel called gene as a factor. They name as a factor. There are some factors which are transferred from one cell to another cell and they carry the information. And Mendel said that substance is a factor. So that factor is now known as a gene. So gene is also known as a factor in Mendel language. Okay. Gene is which is a segment of DNA. What is gene? Gene is nothing but it is a segment of DNA. This is a DNA and gene is a segment of this DNA. Right? This is called as the gene. That is specifying for a particular polypeptide. Right? This is a DNA. This DNA converted into mRNA. After this mRNA, what code this mRNA? This mRNA code for a polypeptide. Right? So this all depend on a gene, that is DNA. Right? So DNA having two parts. One is intron and another is exon. Intron is a non-coding part of a gene. But in bacteria maximum part are a coding part. Means maximum part are a exon part. In eukaryotes, they having maximum part is a intron. Means they having a non-coding sequence. Right? Okay. So exon means coding sequence. Coding part of a DNA is known as a exon. Suppose this is a fragment of DNA. This is a fragment of DNA and ATGC, ATGC, ATGC. Okay. This part is coding part. This is a non-coding part. Means this is a intron. This part is intron. This part is exon. And this part is intron. So polypeptide is synthesized from only this coding part, not by this non-coding part. Okay. So polypeptide synthesis depend on a coding part, that is exon. Bacterial genetics is used as a model to understand DNA replication, genetic character, their changes and transfer to the next generation. Because bacteria having a smallest, it is a prokaryote, right? Bacteria is a prokaryote and their multiplication rate is very high, right? their multiplication rate of prokaryote is high compared to eukaryote. So we can easily see the process of replication, right? Genetic character and how they transfer from one generation to next generation. So that's why bacterial genetics is act as a model. What? They act as a model, right? So there are types of significance of transfer of gene, right? The significance of the transfer of gene, how they are important. Actually, during the transfer of the gene, they combine with other genes. Means bacteria having also a extra chromosomal DNA that called as a plasmid, right? So our desired gene combine with this and form a recombination. What? Form a recombination. That is the most significant things in a gene transfer, during the gene transfer. In bacteria, gene transfer is not an essential part of the life cycle. When it does takes place, usually only some of the genes of the donor cell are transferred to the recipient cells. Remember this. Suppose this is bacteria and they having their genetics material. This is the donor cell and this is the recipient cell, right? When they transfer their gene, they not transfer whole of their gene. They transfer only some part of their gene, right? Only some part of their gene, and that process is known as a, when they combining of the gene with recipient cells, this process is known as a recombination, and the resulting cell is referred to as a recombinant. This cell is known as recombinant, right? Recipient cell is known as recombinant cells. Okay, understand? What are the type of the gene transfer? Actually, there are three mechanisms how the bacterial genetics, how the bacterial gene will transfer from donor to a recipient cells. First is transformation, second is transduction, and third is conjugation. In transformation, naked piece of the DNA taken by the bacterial cell. They not require any medium and vector. This is a process of transformation. See, this is a plasmid present in environment. This is a bacterial cell. They get this plasmid DNA. This process is known as a transformation, right? Another is transduction. Transduction takes place via a vector. That vector is a virus. Bacterial virus is known as bacteriophage. Remember this. What is bacteriophage? Not confuse that there is a bacteria, means they are bacteria. No, this is a virus. What? Virus. Bacteriophage. This is a phage, means they are virus. Next is conjugation. Conjugation means when two bacteria come contact with each other with the help of pilus. Pilus is a tube that present in a bacterial cell. They act as a conjugation. They help in a conjugation process. See, this is a bacteria one. This is another bacteria. This tube is known as pilus. The genetic material is transferred through this tube, right? Genetic material is transferred via this tube, and this process is known as a conjugation. This process is known as conjugation. And another mechanism is also that is transfection. Actually, in transfection, we induce some genetic material via artificially. These all processes are natural. Transformation, transduction, conjugation, this is a natural process. Right? This is a natural process. But transfection is an artificial process. In transfection, we induce the genetic material inside the cell via electrical or another mechanism. So this is an artificial process. So first we discuss about the transformation. What is the transformation mechanism? Conversion of one genotype to another via the introduction, uptake and the recombination of exogenous DNA. Exogenous DNA means the DNA which present outside. The DNA which present outside is known as exogenous DNA. Okay, clear? Because in transformation, no vector is required, right? No vector is required in transformation. So in this process, exogenous DNA are present. That exogenous DNA are taken by a recipient cells. The phenomena is not limited to bacteria or to prokaryotes. Means transformation will take this in another organism also. What is the exogenous DNA? Exogenous DNA already know that it's the DNA which is present outside the recipient cell is known as a exogenous DNA or foreign DNA. Exogenous DNA is bound, then taken up across the cell membrane. This exogenous DNA is taken up by the cell membrane. Recipient cell must be competent and secrete a competence factor, that is a small protein. What is a competence factor? Competence factor is a small protein that activate 8 to 10 other protein, right? This competence factor activate 8 to 10 other protein that affect the cell wall to recognize and membrane transfer of foreign DNA. Right? Means when suppose this is a recipient cell, right? This is a recipient cell. Here this is a exogenous DNA. This is a exogenous DNA, right? When this bacteria recipient cell see this exogenous DNA, one competence factor, what competence factor will secreted by, competence factor is secreted by this recipient cell, right? After the secretion of the competence cells, 8 to 10 other protein will activate so that this protein help cell wall to recognize this exogenous DNA and accept this exogenous DNA inside them, right? This is the property how the exogenous DNA will be taken by a recipient cells, right? Remember that here is a competence factor, means that is a small protein which help in acceptance of this exogenous DNA. Remember this always. DNA taken into a cell is converted to single-stranded form and then can undergo exchange with its homologous region on the recipient cell chromosome. Exchange occur via classical break and reunion mechanism because the recipient chromosome is often circular. A successful exchange required an even number of crossover. Means what they tell? They tell that when recipient cell take the exogenous DNA inside it, after that this exogenous DNA will go inside the recipient cell and transfer into a single stranded, right? After transferring into a single stranded, they recombine with this recipient chromosome with homologous recombination mechanism. After recombining with this with the recipient chromosome, they cross over with this both and mix with this recipient chromosome and form a recombination. Okay, clear? See the diagram. See the diagram. This is the diagram. See what happened here. This is a transformation mechanism with DNA fragment. This is the exogenous DNA. What? This is a DNA fragment that is exogenous. This is exogenous DNA. This exogenous DNA and this is a bacterial chromosome, right? This bacterial cell secrete a competence factor, that is protein. After secretion of competence factor, 8 to 10 protein or other factor is also released. They help the cell wall to recognize this exogenous DNA. After recognizes the exogenous DNA, this exogenous DNA taken up by the recipient cell, right? After going inside the recipient cell, this exogenous DNA converted into a single stranded. After converting the single stranded, they integration by a non-reciprocal recombination. They integrate into a chromosomal DNA and form a crossing over and form a recombination and go inside the recipient DNA, right? And another process, first is they form recombination or another is degradation. If they are not recombined with this recipient chromosome, they degraded into a pieces, and this process is known as an unsuccessful transfer. And this is when form a recombination, this is a stable transformation, right? This is the process of the transformation. And another transformation process is with a plasmid. Here exogenous DNA is present. Here exogenous plasmid is present. Same process, competence factor will release 8 to 10 other the competence factor release the another help in releasing of another 8 to 10 protein factor that help in recognizing the cell wall to recognize the exogenous DNA plasmid. After recognizing, they get this plasmid inside the cell. But this plasmid not recombined with this chromosomal factor, right? They remain separate inside the bacterial cell. So this is also known as a stable transformation mechanism. Okay. This is a previous slide in which mapping of gene primarily is a function of co-transformation frequency. Remember this, only after when transformation will take place, suppose this is a process of transformation, right? This is a process of transformation. During the transformation process, there are some genes, right? So, which gene will it transfer first, then which gene? So, according to the transformation of the gene process, right? According to the transformation of the gene process, we can map the gene. How the gene will transfer inside the bacterial cell. This is known as the mapping of the gene. Suppose this is strand A gene, B gene, C gene. Right? Suppose C gene will go first, then B gene, then A gene. Then the mapping of the gene is C, B, A. Right? This is known as the mapping of the gene. Clear? Next process is transduction. Actually, in transduction, we use a vector. What? We use vector. And that vector is a virus. The virus which act as vector for a bacteria, that virus is known as a bacteriophage. That vector is known as a bacteriophage. So what is the transduction? Bacterial gene transfer via bacterial virus as a vector. This process is known as a transduction. First, what is the introductory material? First is virulent and temperate phase. Virulent and temperate phase and lysogeny. Lysogeny or lysogenic. Actually, there are two types of cycle takes place during transduction. First is lysogeny and another is lysogenic pathway. Right? So remember this, two pathway will take place, lysogeny and lysogenic. Some temperate or lysogenic phage physically integrated into a bacterial chromosome, integrated inside the bacterial chromosome, where other as can remain autonomous in the bacterial cytoplasm. Both type replicate at the time of bacterial cell replication. Means transfer of donor DNA inside the recipient cell. After the transformation, they act as lysogeny or lysogenic cycle. Both DNA will replicate when bacterial or recipient DNA will replicate. They not replicate individually. Right? There are three types of transduction. First is generalized transduction. Second is specialized transduction, and third is abortive transduction. There are three types of transduction. So first, see how the transduction process will take place. This is a virus that is bacteriophage, right? This is a virus that is bacteriophage. This virus contains a DNA, genetic material, right? So this virus come and attached with bacterial cell. After attaching with bacterial cell, the genetic material of the virus will go inside. What? Go inside the bacterial chromosome. Sorry, bacterial recipient bacterial cell. After that this phase will be go away from this. So what happened here? This is a phase genetic material. This phase genetic material go inside the bacterial cell. After going inside the bacterial cell, they synthesize their protein by using the bacterial cell, right? And they develop inside the bacterial cell. This is a virulent phase. Right? This is a virulent phase. After synthesizing their whole cell body, cell create new phases including phase and host DNA, right? This phase contain this host DNA and their DNA both, right? After synthesizing, their whole cell create, when they whole cell create, they synthesize their DNA, and their DNA both, right? After synthesizing, their whole cell create, when they whole cell create, their DNA both, right? After synthesizing, their whole cell create, when they whole cell create, their DNA both, right? After synthesizing, their whole cell create, when they whole cell create, they disrupt transducing phage insert donor DNA. Okay, they burst and this phage come outside, and this phage contain what? Bacterial genetic material also. This phage then attack a recipient cell. This is a recipient cell. After attacking with a recipient cell, they transfer their genetic material, and this genetic material contain both phage DNA and donor DNA or bacterial cell, right? So this is the process how the bacterial host, this is a host bacterial, so how the host bacterial chromosome will transfer in a recipient cell. After go inside, they form a recombination, recombine with host recipient chromosome and form a recombinant DNA. What? This is a rDNA, recombinant DNA. This is a process how the virus help in transferring the DNA material, genetic material. First process, phage inject DNA. Phage enzyme break down the host DNA. Cell creates new phage including phage and host DNA. Transducing phage insert donor DNA. Donor DNA included in recipient chromosome due to process of recombination and form a recombinant DNA. Right? Clear this process? Now we study individually what is generalized transduction, what is a specialized transduction, and what is a abortive transduction. So first, what is the generalized transduction? Mistaken packaging mechanism whereby any piece of the bacterial chromosome vector from bacterial cell to bacterial cell during the lytic cycle of virus. What? During the lytic cycle of a, remember this, during the lytic cycle of virus. Can involve both virulent and lysogenic virus. Any bacterial gene theoretically can be transduced in this way. Each gene has an equal probability of being transduced. Remember this, by this process only low number of transducing particle is produced per infection. What? Only low number of transducing particle will be introduced. During mapping, already I told how the mapping will be takes place. During mapping, that in term of co-transduction frequency, the average size of the transduced bacterial DNA is about 1 to 2% of the bacterial genome. What? 1 to 2% of the bacterial genome can also do classical 3-factor mapping. What? Classical 3-factor mapping. This is the process of the generalized transduction. Generalized transduction will take place by a lysogenic cycle. This is a viral, right? This is the virus that contains genetic material. This contain a viral genome. They transfer their genome inside the bacterial cell, right? What? They transfer their genetic material inside the bacterial cell. This is a phage carrying bacterial. This is a phage. After transferring, they grow inside the cell, and another phage will be formed there. Right? Another phage will be formed there. See, this is the genetic material go inside the cell. This is a viral genome, right? When they go inside this cell, they form a recombination. They synthesize their protein and form a new phage like this. New phage will be formed. After forming new phage, they burst and they go for another cell and transfer their genetic material, right? So, in what happened is, in this donor bacteria were lysed. So, that's why this process is known as a lysogenic lysogenic cycle, right? Lytic cycle. In this, the donor DNA will be burst or they die. Remember this in general. What happened in generalized transduction? This is the process of generalized transduction. Next is specialized transduction. In specialized transduction, improper excision of an integrated prophase occur during the lytic cycle, and a small terminal piece of bacterial DNA is incorporated into the viral chromosome. From name it is clear, means it is a specialized, means special things will be transferred. So in this process, not whole DNA of the recipient will be donor will be transferred into a recipient cell. In recipient cell, a small piece or a special piece of the DNA or gene will be transferred inside the recipient cell. Okay. What happened here? Improper excision of an integrated prophase occur during the lytic cycle, and a small terminal piece of bacterial DNA is incorporated into a viral chromosome. Only bacterial gene on either side of the inserted site can be so transduced, and the transducing particle produced does not carry a full complement of the phase gene. Means they not contain full DNA. Whole DNA will be transferred. No, only a small piece or a small segment of the DNA will be transferred in a specialized transduction. Remember this. In generalized transduction, whole piece of the DNA will be transferred inside the recipient cell. But in a specialized transduction, only a small segment, a small piece will be transferred. Right? See here this is a diagram of a specialized transduction. This is a prophase. This is a bacteriophage, right? This bacteriophage transfer the gene, right? Phase DNA integrated into a chromosome through recombination, right? Prophase deintegrate and pick up a piece of the bacterial chromosome. What happened? Prophase deintegrate and a small piece of the DNA they takes place. Means this is a prophase. They combine of one small piece of the donor DNA. See, this is a donor DNA, right? So they take a small part of the donor DNA. After recombination, the replication of the viral DNA and destruction of the bacterial DNA. Bacterial DNA destruct and their recombination or replication of the viral DNA will be takes place. After replication, whole protein will be formed and new phase will be formed. After forming of phase, lysis of bacterial phase release infection of new bacterial cell. After infection, crossover and stable gene transfer into a recipient cell or chromosome. Okay, this is the process. What happened here? A small piece of the recipient bacterial cell will be transferred here. Okay, clear? Next is the abortive transduction. What happened in abortive transformation? In abortive transformation, bacterial gene vectored from one bacterial cell to another fail to become incorporated in the recipient cell chromosome via crossing over. This can occur with either generalized or specialized transduction. Actually, abortive transduction is not another type of transduction. It is same as a generalized or a specialized transduction. Sometimes what happen? The transfer of the gene will not take place, or crossing over or recombination will not take place with a donor DNA with the recipient DNA. So in that case, abortive transduction will be takes place. Means proper transduction will be not takes place, that is known as, or transduction process will be fail, that process is known as abortive. That process is known as abortive, when transduction process will be failed. It is not another type of transduction. Okay, clear this? What is abortive transduction? Next process is a conjugation. What is a conjugation? Conjugation is also a process of genetic exchange. In this process, gene exchange between sexually differentiated bacteria type, that is plasmid and episome. Plasmid, what is plasmid? First of all, plasmid is an extra chromosomal DNA. What? Plasmid is an extra chromosomal DNA. Extra chromosomal DNA that contain extranuclear genetic element. Episome is an autonomous and or integrated extranuclear element. Episome is an extranuclear element, and plasmid is the extranuclear genetic element. Conjugative plasmid or episome capable of causing transfer of genetic material from one bacterial cell to another bacterial cell. In non-conjugative plasmid episome not capable of causing transfer of genetic material from one bacterial cell to another. From conjugative, it is clear that conjugation will take place, and if conjugation will take place, means there is a transfer of the genetic material. Non-conjugative means there is not transfer of conjugation process will be takes place. So that is non-conjugative, right? This is the process of the bacterial conjugation. How the conjugation process will take place. This is a F plus. One thing also. What is F plus? F plus, there is a F factor that will present in the, this is a F plasmid or F factor, right? This F plasmid present in a recipient, in donor cell, right? F minus means this F plasmid will not present. These are a recipient cell. What? These are the recipient cell. So this F plus will go inside the F minus. How? With the conjugation pilus, with the help of conjugation pilus, right? So this pilus will be formed. They combine with each other. After that, this F plus plasmid will go inside the recipient cell and they replicate, right? And after going inside the F minus cell will convert it into F plus cell. Donor cell attached to a recipient cell with its pilus. The pilus draw the cell together. The cell contact one another. One strand of the plasmid DNA transfer to the recipient DNA, means F plasmid DNA will transfer inside the recipient DNA. The recipient synthesizes a complementary strand to become an F plus. The donor synthesis a complementary strand, restoring its complete plasmid. Clear? Second is what is the F factor? What is the F factor? F factor is nothing, it is a F plasmid. The bacteria which contain F plasmid is known as a F plus. F factor gene promotes cell to cell contact and transfer of the F episome. Both the F factor and the bacterial chromosome contain the insertional sequence. What? They contain insertional sequence. This is the F factor. They contain insertional sequence. These are the relatively small transposable DNA sequence that contain recognition sites, right? And gene coding for a transposon. What is a HFR? When F plasmid combined with bacterial DNA, that is known as a HFR. That is known as HFR. Okay? Clear? Third is F promotes its own transfer from F plus to F minus. Already I told you, occasionally F factor may integrated into a host chromosome and become an HFR. Already I told you how the HFR will become when F plasmid integrated with the host chromosome, then that is known as HFR, high frequency recombination. Right? Clear? Next, what is how the mapping will takes place? Already I told, when the according to the gene, which gene will transfer first, according to that mapping will be takes place. Suppose in this what happened, this is the HFR, this is F minus, this A plus gene, B plus gene, C plus gene. A plus gene will go first, means this is for A, after B this is a C. This is a gene mapping. Okay, this is a gene mapping. Another is sex reduction. Transfer of bacterial gene by a F factor. This process is known as a sex reduction. That occur in a also known as, this process also present in a conjugation. Occur when a HFR return to the cytoplasm but accidentally includes some bacterial DNA that was adjacent to the integration site of the F factor during its stay in the host chromosome. It's nothing but when F plus transfer into a F minus cell, this is process known as a transduction. Right? This is a donor F plus. This is the donor F plus, this is recipient F minus, this is go inside the recipient cell, this also become a F plus, this process is known as a transduction. What is the difference between conjugation, transformation and transduction? This slides contain that. Conjugation, the process of transfer of DNA from donor bacteria to the recipient bacteria. Actually these all are the same, transfer of the DNA. In transformation, exogenous DNA will be taken by a recipient, say in transduction via virus. In conjugation, 2 bacteria will be involved. In transformation, 1 bacteria will be involved. In transformation also 2 bacteria. This process involves physical contact. In this also no physical contact, right? The conjugation process is resistant to DNA as DNA is present inside the bacterial cell. Transformation of DNA, transformation is sensitive to DNA and this is no naked DNA present. So this is a DNA resistant. Okay, clear? So this is all about the bacterial genetics. Hope this topic is clear. Okay, thank you.